To keep it simple, microbial adhesion, biofilm formation, and biofouling. These are the giant Goliaths of desalination membrane. But will a small nanomaterial like graphene bring solution to all these problems? Well, this talk will lead you into graphene world with contrasting hydrophobicities and their putative role in biofilm prevention. Every story has a protagonist and an antagonist, like David and Goliath. The main focus of our study is laser-induced graphene. The sample would look like this. The methodology to fabricate this sample is fairly simple. Supposing if a piece of wood is burnt, it turns into ash. In the same way, if thin film polyimide is burnt with laser, it turns out to be laser-induced graphene. And the fabrication can be done in a laser system which would look like this. Now, this fabrication can be done in controlled atmospheres with particular gases. For example, an inert gas can be passed through this inlet and it can be let out, while within the chamber polyimide is inscribed to form laser-induced graphene. So we have fabricated 4 different graphene types. For example, argon and hydrogen in box and atmospheres containing oxygen like air and oxygen box. So the main objectives of our study are to elucidate the surface, bactericidal and anti-biofouling properties of laser-induced graphene. Okay, the sample is 1 centimeter square and we characterize them for these 3 properties. And the methodology involves surface contact angle and X-ray photoelectron spectroscopy before we characterize our bacteriological studies. Then we employed contact killing and scanning electron micrograph for studying the bactericidal properties of laser-induced graphene. Thirdly, biofilm assay and confocal laser scanning microscopy is done in order to study whether graphene prevents biofilm or not. The bacteria sample which we used is Pseudomonas aeruginosa. This is a model organism for biofilm studies and it is widely known as E. coli of membrane studies or membrane biofilm studies. So, the results for contact killing, contact angle. Now, polyimide is our control, the base polymer. We included another control, graphite. It is because polyimide is a thin film, like a plastic, whereas all the graphene types are carbonaceous. Therefore, graphite will be an appropriate control. So 2 controls and 4 study samples. The contact angle for our control samples are hydrophilic, and these images display the degree at which water droplet interacts with the surface. And if contact angle is less than 90, it is hydrophilic. If it is more than 90, it's hydrophobic. If it is around 90, they are partially wettable. Now, the graphene which is fabricated under inert atmosphere like argon and then in hydrogen box is super hydrophobic. And as you see in this picture, the water droplet just stays above the surface, displaying a contact angle of more than 90 degrees. Now, LIG fabricated in air is fairly hydrophobic, and LIG fabricated in atmosphere containing oxygen is hydrophilic. As you see in the picture, the water droplet almost interacts with the surface. Now, in order to contemplate the reasons on why these contrasting hydrophobicities occur, we studied the oxygen content of the samples using X-ray photoelectron spectroscopy. We found that these two samples, hydrophobic samples, have lesser quantities of oxygen, and LIG that is fabricated in oxygen atmosphere is extremely hydrophilic because they have about 9% of oxygen. Even though LIG air is hydrophobic, it contains a higher degree of oxygen when compared to these two samples. Why? Okay, it is because recently it has been found that graphene that is fabricated under nitrogenous atmosphere is super hydrophobic. Now you have the answer. Okay, atmosphere contains 78% nitrogen. That makes this sample fairly hydrophobic, even though it has a larger degree of oxygen. Now we will see a small movie in which in which the water droplet does not want to stay on the super hydrophobic surface, but it wants to jump to the hydrophilic polyimide. So now the contact angle and X-ray photoelectron spectroscopy studies of laser-induced graphene is a done deal. We will move on to the bactericidal properties of laser-induced graphene, which is studied using two methodologies, namely contact killing and scanning electron microscopy. So when we did contact killing, we looked for certain amount of viable cells after the bacteria is incubated with graphene surfaces. We see that the number of viable cells for our control sample, which is polyimide and graphite, is about 140. CFUs refers to colony forming units, the number of bacteria. So polyimide and graphite, after incubating with bacteria, shows about 140 colony forming units. Okay, so we have to find out whether graphene kills bacteria or not, and we found that they have 60 to 76% inactivation. Well, in order to prove that this is not just inactivation of bacteria, but it is really bactericidal, we did scanning electron microscopy. And as you see that the scanning electron microscopy for polyimide displays healthy bacterial cells, whereas graphene sample displays cells of compromised integrity and the cells are flat. Now we have proof that graphene is bactericidal. So things are wrapped up with surface and bactericidal properties of laser-induced graphene. Now we will move on to the major component of our study, which is anti-biofouling properties of laser-induced graphene. Now we did this using flow cell experiment and confocal laser scanning microscopy, and then the biofilm is quantified, and the bacterial species is Pseudomonas aeruginosa. Before going on into the bacterial studies, we will just look into the background for bacterial adhesion and biofilm formation. Now, the components of biofilm we studied are dead biofilm, live bacteria, which secretes extracellular polymeric substances, which comprises of proteins and carbohydrates. Now, take for example a sample, say our control polyimide. When it is flowed through with bacteria and nutrients, bacteria adheres to it. Once bacterial adhesion happens on polyimide, their genes are triggered to secrete this extracellular polymeric substances. Therefore, bacteria are attached to each other and then they are adhered to the surface. Now, when bacterial colony forms like this on surface, it is called as biofilm. And when bacterial biofilm damages the membrane, it is called biofouling. Okay, next time I ask question. answer, okay? Now, laser-induced graphene is bactericidal. So, what do you expect it to be? No, it's anti-biofouling. Nevertheless, it is just incubation of graphene with bacteria. But what happens if we flow bacteria through laser-induced graphene for 36 hours? Any guess? As we pass bacterial cells through laser-induced graphene, there are certain cells which are capable of surviving, and they are known as persistor cells. And these persistor cells are capable of secreting extracellular polymeric substances, and which is known as EPS conditioning. Now we know that our antagonist is really strong because they are capable of resisting the protagonist. So now it gets thrilling. Persistor cells that secrete EPS covers the surface that is bactericidal. Therefore, more bacteria grows on the surface and biofilm forms. This is what we have expected, but we found in our results that a plethora of biofilm component comprises polyimide and graphite, except that graphite is lacking some dead biofilm components, while laser-induced graphene that is fabricated under inert organ atmosphere and in hydrogen bonds has extracellular polymeric adhesion on it. Now, it is because these samples are hydrophobic, which means they could repel water and they could attract organic components. And graphene is capable of removing organic contaminants from wastewater. So this could be a possible reason for EPS adhesion on these two samples. One more point to add, the biofilm is more dynamic than we think. But that's not the case here, because these samples are hydrophobic. So even though the EPS grows larger or thicker, the hydrophobicity of these two samples withholds the extracellular components towards its surface. So hydrophobic samples containing sporadic amount of live biofilm components and a lot of EPS. When compared to these two samples, the dead biofilm, the live, and the polymeric substances they secrete are nothing but a drop in the bucket for hydrophilic laser-induced graphene samples. Now, before our closure, we recapitulate. Graphene that is fabricated under inert atmosphere is hydrophobic, and graphene that is fabricated in oxygen atmosphere is hydrophilic, and graphene is bactericidal. So, and anti-biofilm properties we studied and we found they are quite anti-biofouling against Pseudomonas aeruginosa. But to sweeten the deal, we want to scrutinize okay, this is a gram-negative bacteria, and to make it more interesting, we have to scrutinize the anti-biofouling properties of laser-induced graphene against a gram-positive bacteria named Enterococcus faecalis. We will look into the difference now. Gram-positive bacteria has a thick layer of peptidoglycan around their plasma membrane, whereas gram-negative bacteria has a thinner layer followed by outer membrane. Now, there are evidences in literature which states that a nano-material is capable of puncturing through the thick peptidoglycan layer, which causes the bacterial fluid components to lyse out. So, in order to test whether our sample would prevent gram-positive biofilm or not, we repeated the same test using confocal laser scanning microscopy, and we quantified the biofilm components. The results are almost same, and the graphene that are fabricated under inert atmosphere and hydrogen bonds has EPS adhesion, extracellular polymeric adhesion, while these two samples holds no account for biofilm adhesion. So, having behind us a bright future with graphene, we will bring back to our mind their nature in the realm of microbiology and surface chemistry. The increase in oxygen contents make it hydrophilic, and graphene is killing bacteria. And assuredly, they are anti-biofouling for both gram-positive and gram-negative bacteria, except for some polymeric adhesions. The plausible mode of action for biofilm prevention is because it is bactericidal. So, advantages and disadvantages. We are reaching the end of line with words of wisdom said by a scientist. Pasteur once said that, "Gentlemen, these are the microbes which have the last words. Verily, verily, bacteria are capable of growing in extreme environments, and they have developed resistance against antibiotics. But will a nanomaterial, possibly much smaller than graphene, have last words? Researchers, what's on your mind?" Thank you.
All right, we have time for question two. Yes.
Kim.
I find it interesting that hydrophilic are better anti-fouling than hydrophobic, since the E. coli, which are negative, gram-negative bacteria, have this RPG outer membrane, which is charged negatively by phosphate groups, if I'm not mistaken. So why hydrophilic?
Both the gram-positive and gram-negative bacteria is fairly negatively charged. So it doesn't matter whether the graphene is hydrophobic or hydrophilic. Like EPS is an organic component. And we saw a large amount of EPS secreted, and a bacteria is not capable of secreting that much amount of EPS. Possibly because that sample is hydrophobic, they absorbed some glucose, not glucose, mannose from the solution. So when it comes to anti-biofouling, both hydrophilic and hydrophobicity works. It's just the bactericidal property of graphene that is involved in anti-biofouling.
All right. Any more questions? No. So let's thank the speaker again.